target sites. Type 2 restriction endonucleases, they can recognize a particular target site and cut within it. So these type 2 restriction enzyme, they can cut those recognition site that is having a rotational symmetry. Simply we can say that they recognize those nucleotide sequences at specific site. Either it may be 4 base pair or 3 base pair long or 6 base pair long, that is having rotational symmetry or palindromic sequences. Now what is palindrome? Palindrome means that the words that are read similar either from the right or the left side. For example, here this is the recognition sequence for EcoR1 restriction enzyme. So which sequence we can call it as a palindrome that read the same either from 5' to 3' or 5' to 3'. So here GAATTC, it is from 5' to 3' in one direction, and then it will read the same from another direction, like same reading but from another side, that is GAATTC. So such sequences, they are called as palindromes. And in the next, the cutting site has been indicated by the forward slash. So these are the cutting site where EcoR1 restriction enzyme, it can cut the DNA. And these asterisks, they indicate the position where adenine can be methylated by separate methylase activity of EcoR1. When EcoR1 cut the DNA, then it can produce single-stranded overhangs, that is, single strands that are protruding at 5' ends. So it is the generation of cohesive end. Cohesive end are the single-stranded DNA whenever they came in contact with some other DNA source, then the base parent it is there, so that after annealing the gaps can be bridged by the DNA ligase. So this is the example of EcoR1, that how it can cut a particular recognition site or sequence, and here this sequence it is composed of 6 base pairs. Then there is another example of different enzymes that can cut different recognition sequences. Here it is the example of He3. It can recognize a DNA sequence that is composed of 4 base pairs. So mostly a recognition sequence, for the sake of convenience, it can be represented by even by a single strand like 5' to 3'. So He3 recognize GGCC, and when it cut the DNA from here, this is the recognition site, so it can produce the blunt end. Blunt end means that there are no single-stranded protruding or overhangs, like in case of Ph.D. This is another example of restriction enzyme that can recognize 6 nucleotide sequence, and when it is cutting, it can produce sticky end with 3' protruding or overhangs. And then that we have discussed, it is EcoR1. It is also producing sticky end, but the fragments or the termini that are produced, they are having 5' overhangs. So the DNA that can be attacked by restriction enzyme, they can produce different type of termini. So here and other examples are there, like BAMH1. It can recognize 6 base pair sequence, and steric indicates the site of methylation. Then some enzymes can recognize 8 base pair sequence like NOT1. So a different type of restriction endonucleases are there that can recognize different target sequences and produce different fragments. This is another example of interaction of the DNA from different sources. So this example, it is the DNA joining from different sources. And this is the example of DNA that has been digested or fragmented with EcoR1, so that single-stranded cohesive ends are there that are represented by AATT. And when DNA from another source, if it is cut with the same restriction enzyme, that is EcoR1, it is having the same sticky ends that are represented by AATT. When they are mixed together, then they can be ligated. And these nicks or joints can be filled by DNA ligase. Now, sometimes what happens, molecules can join after fragmentation by EcoR1. So this is another example, intramolecular association. Suppose we have cut a DNA, a fragment like the vector with the EcoR1. So two fragments may be there, and sometime what happens that they can reassociate in the absence of foreign DNA. So this may be the situation that sometime transformants having the foreign DNA and sometime intramolecular association is there where no foreign DNA it is there. So from this discussion we can say that restriction enzymes may be of different types and they can recognize different recognition sequences, and even the fragments that they are produced, they can also vary in lengths.